the first talk, which is from Roozbeh Farhoodi. He's a postdoc researcher from Conrad Cording's lab at UPenn. And the topic of his talk is the dendrograms of neuron morphologies exhibit asymmetrical motifs. Okay, Roozbeh, do you want to take the lead? Okay, cool,
awesome.
Great. Thanks.
Do you see my screen now?
Yeah, cool. We can see
okay,
that.
Awesome. Yeah, thank you so much, Anqi, for introducing me.
Sorry, Roozbeh, we see it as a presenter mode, not.
Oh,
sorry.
I see. Give me, I'm sorry. Okay, so I have to stop this one. That was my mistake. I have to share the other mirror. I have a mirror one. Do you see it now? Is that working?
Mode.
Is it still in presenting mode?
The rehearsal mode, like with 2 slides.
Oh, I see. Okay, so wait. Okay, so I hope that is this one working. Let's see. It's good?
Cool. Yeah, that's good.
Great. Thank you so much again. I'm sorry for that. Yeah, so I am going to talk about the dendrogram of neuron morphologies that exhibits asymmetric motifs. Before starting, it's more than a century ago that Ramon Cajal opened our eyes to neuron morphologies. He drew all of these pictures by his hand, like staining neurons that he did in his lab and look at on the microscope. And now, a century after that, we have a bunch of tools and techniques to image from very close resolution to nanometer resolution. And we even are able to picture the whole brain connectome. And that has opened the door for us to study the structure, the geometrical structure of the brain. Okay, so what is the neuron morphologies? If you haven't heard about neuron morphologies, I have actually plotted a few of them, 6 different cell types here that are, for example, from omicron cells that are in the retina cell, in the retinal area of the brain. These are all from the mouse brain, or perconjugranal cell from cerebellum that are working on the motor system, or different pyramidal cell in different layers of neocortex. Even we have a good converged way of like putting all data together, and one of the good successful one is neuromorpho that has more than 100,000 neurons of 50 different species. So we are able to look at the variety of the cells in different brain regions and species. In my opinion, there are 2 main questions when we see neuromorphologies. The first one is that how we can describe this geometrical structure. What is their geometrical feature? What generative models make them? And this is kind of a question that relates to the cell type identification. If we want to find, for example, new cell types, we have to say, based on the morphology, of course, we have to say that what kind of geometrical feature we expect, or developmental studies that is the neuron is changing over time, or even larger scale simulation of the brain that we want to make a big like simulated version of the brain. And the second question is that given the morphologies, what kind of function we expect? In other words, what kind of what kind of function we can see when we have this geometrical structure? This is a question that relates to the physiology of neurons. We know that the simple sigmoid function of neuron is not enough to describe the function, but then what is the best way to describe them? Or the other thing that is related is the biological plausible neural networks, how we can incorporate these morphologies in the neural networks. Okay, so let's go from the morphology to a little bit simpler structure. Here I have plotted one neuron that's connected possibly to a few other neurons in the synapse with different synapse locations. And in the dendrogram structure of this morphology, I ignore all the geometrical structure and just keep the branching order. For example, I would start from soma and see that where is branching and add a branching to this diagram here. Even I can see that where are these connections or where are the synapses on top of that. Okay, so it's a simpler version of the morphology. We are ignoring the all 3D structure of that. But still, it's not very simple to study. Here I have another like Purkinje cell, and its dendrogram that is sorted different based on the depths of the distant dendrogram. We can use, for example, a simple motif analysis to see that within this graph, what kind of branching we see. For example, here I can have 3 different branchings. The branchings that both of the children that it has are leaf, like for example, all the branchings around the end of these 3, or the branchings that one of them is leaf, the other one is branching, and the branching that both of them, like the red one, both of them are branching. And you can count the number of them with different depth and see that how this profile changed from one cell to another cell type. This is, for example, for the Purkinje cell. You can do for the bigger motifs, these are the motifs I will call them, and you can sort them and kind of differentiate different cell types. For example, for a few cell types, for apical dendrites, basal dendrite, granule cells, ganglion cells, we can count that how many motifs of order 8, by order 8, I mean 8 roots, 8 leaves at the ends, how many of them we see in the single neuron, or if we average it on average, how many of them do you see that? And you can see this trend to differentiate different cell types and see that are these 2 classes different or not. But there is a big challenge when you do this analysis. First of all, it's a simple analysis. It doesn't give you good intuition, but the main challenge is that this number of motifs, if you a little bit go further than 3-4 nodes, they grow super exponentially. For example, if you have 4 nodes here, you just have 2 different topologically different trees. If you have 5, you have 3 different ones. But if as soon as you go to the higher and higher number of leaves, you have more and more number of motifs that you can generate, and it doesn't give you a good way to sort them. Okay, so here in this talk, I want to give you a simple model that describes the morphologies. Let us start with the simplest branching process that we can think of. In the simple branching process, you have a probability distribution, let's say P, and based on the P, each node it may branch or terminates. And this probability P can depend on the depths that you go. This is, for example, one typical example. If you have the P is equal to 0.045, and even you can, given this P, you can look at the likelihood. This is a generative model, so we can compute the likelihood, and the likelihood only depends on the branching and leaves. That's a very simple model, simplest models that we can think of. But we know that's lacking a bunch of information. For example, if you look at the neurons, in most of the case, you see that there is a main branch that's coming out of soma or somewhere, and then there are some small side branch that's coming out in different locations. If you see under developments, you see that this main branch that are usually thicker, they stay, and they just like have small things coming out, retract, and they continue growing. Okay, I want to add this feature to the simple model that we have. It's actually very easy. I'm going to extend the branching process that I showed you. Let's assume that we have 2 different types, let's say A and B. A correspond to the main branch and B correspond to side branch. And you have 2 different probability distribution that possibly these PA and PB are different, that if you are in the A location, A node, you have the probability PA to branch, and if you are in the B nodes, you have the probability PB to branch. If PA is very high, for example, in the case that you are in the main branch that's wants to continue, and PB is very low, you have a typical structure like this one, that the main branch is trying to survive and the side one is dying as soon as it comes up. Okay, great. So after we wrote this model, it's easy to generate the samples here. But one hard problem that we had was that given this model, how we can calculate the likelihood of each tree? Like given this tree, for example, and P and PB, what is the likelihood of seeing that? After a bunch of try and error, we found the formula in this case. And the formula that is here, if you look close at this formula, you see that there are a few more parameters. n plus minus, n plus plus, and n minus minus, that if I flash back to other slide that I showed you, this one, this is actually the number of these values in different locations. Okay, great. So we have now a model, we have a generative model, and we have a likelihood of them. Then next is to try in different cell types, see that what we observe from different cell types by applying these models. Okay, so fitting on the cell types, we observe that that is actually a very typical structure that when we try different cell types, we observe that for example, let's say in apical dendrites, you have a point that P and PB, these are a graph, the blue one is the PA, the orange one is PB, and this green one is the simplest model that I presented at first. And I'm fitting on different depths. Okay, so when I apply on, for example, this subclass, the apical dendrites, for example, I see that at some point, they fork and PA become higher and higher, PB is become lower line. It means that in this area, the neuron is very asymmetric compared to this area that's probably very symmetric growth. And this pattern repeats almost in all the data set that we had. That was interesting for us. That is a general phenomenon. Okay, great. So we have a thing, we have a model, we have fitted on the cell types to see some profile of the parameters in these models, but how we know that these models are superior? We have likelihood, and this likelihood tells us to compare different models. For example, for this case, we can simply compare them with the symmetric one that has one parameter, or the asymmetric one, P and PB that I told you, the one that are depth dependent, or the one that are not depth dependent. And the general message that we learned was that that's actually true, that the asymmetric one is actually superior when you fit on the dendrogram. Great, so this is the end slide. I'm going to just give you takeaways. We have three different takeaways here. One is that we can use motifs to compare different cell types. And this is a way to distinguish different cell types or possibly find the new cell types. We present a depth-dependent asymmetric models, and we observe that superior to other models. And when we feed, we realize that at the beginning, they are very symmetric structure, and in the longer scale, they have very asymmetric. And thank you so much for joining us. I want to acknowledge a few people that helped me for these projects. Konrad Kording, that he's a PI, and as you see, he's very much into the CPUs and Neuron Morphologies. David Rolnick, he's right now in the Professor and Mila, and that is basically the work with him. Khashayar, we have worked on the functional part that I haven't talked about that. If you have time, we can go through that. Lena, that she's actually presenting a talk very soon after here, and Devang and the rest of the coordinating legs. Thank you so much, and I am here for any question that you have.
Great.
I don't know how much time we have.
We have 1 minute-ish left. Thanks, Roozbeh, for the great talk on this interesting analysis with the dendrogram of the neurons. One Q&A question we have is, how do you encode the depth? How do you encode the depth dependence on the model? To further clarify, is there some sort of functional form for how PA and PP change with depth?
That is very good question. Thank you so much. Okay, so I had actually two bonus. I can go quickly to these two bonus. One of the aims that I have is that to simplify the structure of neurons. For example, if you have this one, you can say, okay, that's a different compartment that it has. And this comes back to the structure that I told you. If you have this simple structure, one observation that we had, and that was a thing that still we are studying, that is that you can try different structure, for example, trees that, for example, different trees with different networks that are associated with each nodes, and see that how well they perform on different tasks, like let's say CFAR task. And the thing that we observe, and that's kind of our counterintuitive, is that, for example, for CFAR tasks or for image classification tasks, when you have more symmetric one, you have a better chance that you train on the data set than you have asymmetric one. It's a kind of reverse, like fix that. But like, that's a question that if you have different structure, what kind of data set it fits better? That is actually a question that we are still studying on that. There is actually a next talk after here, Elena. She's talking about the similar stuff if we are interested in that.
Great. Thanks for the question. Thanks for the answer.